I would suggest that you draw yourself an epidermis too. It doesn't have to be as pretty as mine's going to be, but the actual drawing of it is what helps you. So maybe you want to watch me draw it this first time and then rewind me, draw your own, or come back later and draw your own, something like that. So if you want to watch me draw it, that's perfectly fine. I'm going to begin with what? Well, these are dermal papillae, aren't they? Sure. Papilla, singular, right? So the dermal papillae. What's below that? Well, a bunch of connective tissues, some collagen, some elastin, dense, beautiful fibers down here. I'll be like, what's his name, Bob Rossi. These are happy fibers. This is where they live. You put your fibers wherever you want fibers. So here's some connective tissue fibers. This is the dermis. I don't care what's down there. So here we have dermis. And then above we have the epidermis. And this will take me some time, but that's okay. I've got the time. That's what I'm here for. So I'm going to draw one layer of cells. These would be keratinocytes. Right here. And they tend to be sort of cube shaped. So I'll draw some cuboidal cells here using my fancy tablet. Maybe this will be a little faster, to give me colors so it's easy for you to see what's going on here. And I always recommend, I'm a big fan of drawing your own diagram. The drawing of it is what you want. You can find lots of pictures that somebody else made of the epidermis. Those are great, but they don't really work that well as a study tool. What helps you is drawing your own. As I always say, your picture does not have to be as pretty as mine is, but make it as close as you can. And if you think this is pretty, well, I got a bridge in Brooklyn to sell you then. Okay, so here's my basal surface, right? Everybody remember some of these structures from your epithelium discussion in tissues. Bear with me, I'll get there. Okay, so one layer of cube-shaped cells right at the bottom on top of the dermal papillae. And just to keep me honest, because I know that's important for you, don't let me get away with anything here, let's put in our intercellular cement, this gel, proteoglycan, intercellular cement, cell glue, however you want to think of it. That would be right here. Remember, the papillae increase the surface area for adhesion here, and every so often some protein stitches making hemidesmosomes, forming the basement membrane, holding this epithelium down onto its underlayment. If none of this is making sense to you, you need to back up and watch some other videos, because it should. Now, this first layer of the epidermis, so this would be the deepest layer of the epidermis, is called the stratum basale. So the most inferior layer, stratum basale. This is one layer of cuboidal keratinocytes. One layer of cuboidal keratinocytes. That's the Stratum Basale. Sometimes called the Stratum Germinativum. I'll say that again. You may say this in your textbook depending on what author you have. The Stratum Germinativum. Do you know why it's called that? Because there's lots of mitosis here in the Stratum Basale. Lots of mitosis. This would be totally normal, everyone. If you looked at the Stratum Basale, maybe this cell right here, look at this one, I just put an X in it, maybe this cell's in an obvious state of anaphase in mitosis. Maybe a couple cells farther down the line, this one over here, maybe it's an obvious metaphase. And here's prophase, and here's telophase. So this would be totally normal to see lots of mitosis going on down here because this is the germinating layer of the epidermis. That's why they say basale sometimes. This is where the epidermal cells are being formed. So all the keratinocytes that you're going to see as we move up the strata originated down here in the basale. This is where all the division is happening. The melanocytes would occupy this layer. We'll talk about those later. And you'll also see some of the vitamin D precursor molecules are made in this layer, the Stratum Basale. So one layer, cuboidal keratinocytes, doing lots of mitosis, very active mitosis here. Next up, just using a different color, so it looks different for us, is the second stratum of the epidermis. This one's a good bit thicker, both in number of layers and dimensionally if I was measuring it with an actual ruler. So here I have maybe 8 or 10 layers of keratinocytes. 2, 3, 4, 5, 6, 7. So you sort of get the idea here. I'll fill it in, but I'm just going to go like this and not take all the time because you get the point. Everybody here has a nucleus. Oh yeah, isn't that beautiful? Look at all these happy cells. And again, this is your painting, so you put your cells wherever you want your cells to be. This is where they live. All right, so enough tribute to Bob Rossi. This second layer is the stratum, I'm going to abbreviate here, spinosum, O-S-U-M. The stratum spinosum. Doesn't like it out there in no man's land. Sorry about that. Stratum spinosum is the second layer going up. This is 8 to 10 layers of keratinocytes. There's still some mitosis going on here, and we've now started some keratin production. So I'm color coding my descriptors here. There's still some mitosis that goes on in this stratum. Some mitosis going on here. And keratin production has begun. So maybe, let me highlight a little bit. Maybe this cell right here is still doing mitosis, but this cell right here has started making keratin. It's not really doing mitosis anymore. This is perfectly normal in the stratum spinosum. So this second, moving up, stratum of the epidermis has a lot of cells in it. Some of them, deeper ones, would still be engaged, finishing up their mitosis. And then others are starting to make keratin proteins, that tough, remember, fibrous protein. So it's starting to build up in some of these cells, little keratin fibers that they're making inside the cell. This is not a connective tissue. They're not making keratin fibers and spitting them out the way a fibroblast does with, say, collagen fibers. They're just starting to make some keratins here. So the farther up we go, the more keratin fibers these cells are getting filled with. And the farther down we go, the fewer keratin fibers we have and the more mitosis that's going on. Because the youngest, newest epidermal cells are down here in the stratum basale where all the mitosis is going on. And then as they move up, they're getting older, these cells. Next up, look at your textbook, look at your pictures. What layer do we have? We have what's called the Stratum Granulosum. Right here. Stratum Granulosum. I'm going to make 3 layers of cells here. Stratum Granulosum. That's a G, by the way. Stratum Granulosum. So this is 2 to 5 layers of keratinocytes. And what we have going on here is no more mitosis. We're not really doing mitosis in this layer. The cells are too old for that. We have keratin production going on here. Keratin production and keratohyalin production going on here, and the release of some lipids that happens here. Now, I'm going to zoom in on the bottom where I have a little blank space. I'll come back to my wonderfully artful picture. But let's just go down here where we have a little space. So in the stratum granulosum, we have keratin production, keratohyalin production, and the production and release of certain lipids by these cells, these keratinocytes in this stratum. Keratin, we know, is the basic, tough, water-resistant protein. If I can remember how to spell. Remember, you're not water-proof, you're water-resistant. That makes up a lot of those external coverings we find in biology. Not all, by any means. Keratohyalin, this is a different protein. This is a protein within these cells that appears as dense granules, and it links keratin fibers together. So let's take a look at a keratinocyte, say, of the granulosum, which I think I made orange. Yes, I did. Okay. So let's say I have a keratinocyte right here. I'm flattening out a little bit. It's not looking quite so cuboidal anymore. And inside this cell, I have these keratin fibers, right? And then I have keratohyalin proteins that come in and link these things to one another. Forming them into what we see as these dense granules inside the cell. While at the same time, these cells are releasing small amounts of lipids, you know, fats, oils, waxes, lipids, onto the surface of the cell. Remember the thing about lipids? They don't get along with water. So if I come back up to my Stratum Granulosum, its name comes from the fact that I start seeing these granules forming in these cells, which are the linked up keratin globs. So look close, I'll pause for a moment, look close at the micrograph you might have in your textbook by whatever author. The way we know the granulosum is because it has all these granules in it, which are caused by the linking of keratins by keratohyalin proteins. So it looks all grainy, spotty, dotty, dark, dense. And the fact that I'm putting lipids on the surface of these cells, let me try a tiny, tiny highlighter maybe. So let's just take this cell right here. Lipids all over the surface. And do you remember how lipids get along with water? They don't at all. So that means no water in, no water out. The cell starts to dehydrate. It's in this stratum right here. This is where the keratinocytes die. This is where they die. Right here in this stratum. If it had a little sound, you might hear, this is water sucking noise I'm trying to make, as these cells actually dehydrate and die. To put it a little more bluntly, in your typical epidermis, Stratum Basale, everybody's alive, actively doing mitosis. Stratum Spinosum, whoa, everybody's alive. And what are they doing here? Mitosis a little bit, the deeper we are, and a little bit of keratin production. But in the Stratum Granulosum, everybody dies. Cells enter the Stratum Spinosum alive, and then by the time they're here, they're dead. This is where they die. The keratinocyte graveyard, if you will. So everything above this layer or superficial to this layer is dead. These cells are dead. So I would have living keratinocytes in the basale and the spinosum, obviously, and then I would have living, dying, and dead keratinocytes in the granulosum, but everybody above that, plain and simple, would be dead. But there still are a couple strata above here. You can look in your textbook, any micrograph will show you there are still two strata we have yet to describe. So next up, let me do a little erasure here. Whoa, that's a lot. That was more than just a little. But let's see if I can undo that. Save us a little time. I have no idea why my software wants to keep jumping down. That must be where my cursor left off. But, okay. So we have another strata working our way up, which is the Stratum Lucidum. So next up, we have the Stratum Lucidum. Now these would all be dead cells in here because everybody above the spinosum is dead. And they're very flat here. So this stratum that we see right here is the Stratum Lucidum. U-M. That's a D. Stratum Lucidum. Just a few layers. You know, maybe 4 or 6. 4 to 6 layers of flat, dead, clear keratinocytes. Flat, dead, clear keratinocytes. Everybody's dead. Everybody's flat. And the cells tend to be extremely clear in appearance. Look at the micrograph you see in your textbook. Look at the micrographs you see in lab. Look at my Lab 2 lesson. This would be that layer that is clear. It's sort of a landmark for you to use in lab. I always tell people to look for the granulosum, grainy, dark layer. That's a good landmark. And if you can't find that, you might find the lucidum. Now notice I say might. Pay attention to your lecture upcoming. Not everywhere has lucidum. Not all of you has this stratum. This one is only found in what's called thick skin, which is kind of where we'll pick up next time we meet. But before we leave, let's finish off our epidermis with the last epidermal layer. How about plum for that one? This is the Stratum Corneum. Stratum Corneum. I'm getting sick of drawing keratinocytes already, but I think you get the point. This is the Stratum Corneum. Highly variable as to how thick it is, and I'll just write right here, right on it. This is the Stratum Corneum. This is the part of the epidermis that we see when we look at each other. It would range in some parts of your body to as low as 15 layers all the way up to about 30 layers in some parts of you. Of what? Dead bags of keratin. 15 to 30 layers of keratinocytes. I use that term loosely here because really what we're talking about are squamous, dead bags of keratin at this point. That's what we have, the Stratum Corneum. So in some parts of us, it's thinner, maybe 15 layers. Other parts where your skin is tougher, say calluses, things like that, you might have as many as 30 or even more in some areas, layers of keratinocytes here, but they're all dead. So the lucidum, the corneum, everybody's dead. Looking at this picture, before I leave it, this is just a way, hopefully, now in lecture, it forces me to slow down enough and describe these 5 strata with some notes, and you can write down information that we can then test you on. But as a study method, draw yourself some epidermis several times. Label the strata from basale to spinosum to granulosum to lucidum to corneum, working your way up from deep to superficial, and use it to help you both for your understanding in lecture and your understanding in lab. Again, Watching me do it, I'm sure, is loads of fun, he says sarcastically. Doing it yourself is what helps you understand it and know it for your test. So I strongly encourage you to draw your own epidermis several times. Don't just try to use mine. It's as great as it is. That isn't as helpful as drawing your own with labels describing what happens here. So again, you can rewind me, pause me, slow-mo me if you have the ability, all that sort of stuff if you want to. Going through the epidermis